The pleasure and great opportunity to deliver this lecture on spices and their antioxidant properties at the symposium. I work at Mondelēz International as program manager and associate principal scientist in R&D. I'm joining the symposium from New Jersey. I had the opportunity to work on antioxidants, specifically flavonoids and anthocyanins during my doctoral research, and I'm glad to share the learning in the context of spices. In this presentation, we will cover the following topics. We will start with free radicals and how they cause oxidative stress. This is a good segue to an important topic, antioxidants and what they are. We will take a look at bioactive compounds that have antioxidant potential. Then we will move on to measurement of antioxidant capacity, some basics, reaction mechanisms. And we will review the dietary antioxidants in selected foods and milestones in antioxidant research. This leads us to our main topic of spices, their application, phenolic antioxidants, mode of action, and bioactive compounds. We will also review in detail the antioxidants in selected spices. Before we close, we will take a look at bioavailability, safety, and toxicity as well. Growing knowledge of free radicals and reactive species have revolutionized health and disease management. Free radicals and antioxidants have become commonly used terms in modern discussions of disease mechanisms. Free radicals and reactive species such as reactive oxygen species or reactive nitrogen species are derived either from normal essential metabolic processes in the human body or from external sources such as exposure to X-rays, ozone, cigarette smoking, air pollutants, and industrial chemicals. Any molecular species that contain an unpaired electron in atomic orbital can be called a free radical. They are unstable and quite reactive. This means they can donate an electron or accept an electron from other molecules to achieve stability. Examples of such free radicals are hydroxy radical and superoxide anion. Hydrogen peroxide, hydroxy radical, and superoxide anion are collectively called reactive oxygen species. Free radicals attack important macromolecules such as nucleic acids, lipids, proteins, leading to cell damage and homeostatic disruption. Living organisms possess finely regulated systems to maintain very low levels of free radicals and reactive species. That is, their production and elimination are well balanced. However, under certain circumstances, this balance can be disturbed, and there are several reasons for that. Increased levels of endogenous and exogenous compounds entering autooxidation coupled with ROS production, depletion of reserves of low molecular mass antioxidants, inactivation of antioxidant enzymes are a few. Hence, when there is an imbalance in cellular environment, the result is oxidative stress. Short-term oxidative stress may occur in tissues injured by trauma, infection, heat injury, toxins, and excessive exercise. These injured tissues produce increased radical generating enzymes, activation of phagocytes, release of free ion, copper ions, producing excess ROS. Oxidative stress may contribute to the development of various diseases, including cancer, atherosclerosis, age-related diseases, and neurological diseases such as Parkinson's, Alzheimer's, and depression. So what are antioxidants? An antioxidant is a molecule stable enough to donate an electron to a free radical and neutralize it. This way, it reduces its capacity to damage vital molecules. Some such antioxidants are glutathione, ubiquinol, and uric acid. They are produced during normal metabolism in the body. Other lighter antioxidants are also found in the diet, such as vitamin E, vitamin C, and beta carotene. The antioxidants acting in the defense system act at different levels, such as preventive, radical scavenging, and repair. The first line of defense is the preventive antioxidants, which suppress the formation of free radicals. To suppress such reactions, some antioxidants reduce hydroperoxides and hydrogen peroxide beforehand to alcohol and water. Glutathione peroxidase and glutathione S transferase are examples. The second line of defense is the antioxidants that scavenge the active radicals to suppress chain initiation and or break the chain propagation. Various endogenous radical scavenging antioxidants are known. Some are hydrophilic and some are lipophilic. Vitamin C, albumin, bilirubin, and thiols are hydrophilic. Vitamin E and ubiquinol are lipophilic. The third line of defense is the repair and de novo antioxidants. Proteolytic enzymes, proteinases, proteases, peptidases present in the cytosol and in the mitochondria of mammalian cells recognize, degrade, and remove oxidatively modified proteins and prevent the accumulation of oxidized proteins. Let's take a look at different types of antioxidants. They can be classified as enzymatic or non-enzymatic. Cells are protected against oxidative stress by an interacting network of antioxidant enzymes. Here, the superoxide released by the processes such as oxidative phosphorylation is first converted to hydrogen peroxide and then reduced to water. This detoxification pathway is the result of multiple enzymes, with superoxide dismutase catalyzing the first step and then catalases and various peroxidases removing the hydrogen peroxide. Glutathione systems include glutathione, glutathione reductase, glutathione peroxidase, glutathione transferase. This system is found in animals, plants, and microorganisms. One of the non-enzymatic compounds or antioxidant is vitamin C, which changes to ascorbate by donating an electron to the lipid radical in order to terminate the lipid peroxidation chain reaction. In other words, antioxidants can be grouped as exogenous and endogenous. Exogenous are those which are derived from the diet, like vitamin C and D, for example, and endogenous are made by our body, such as glutathione. Phytochemicals can be defined as bioactive compounds produced by plants, but it may include compounds that may affect health but are not essential nutrients. There are different classes of compounds as shown here that have antioxidant properties like phenolic acids, flavonoids, carotenoids, sterols, tannins, terpenes, and others. Synthetic phenolic antioxidants currently permitted for use in food are BHA, BHT, propyl gallate, and TBHQ. In recent years, the possible toxicity of synthetic antioxidants has been considered. Thus, the potential of plant products to serve as antioxidants has been explored. Methods and tools used for measuring antioxidant activity has advanced remarkably during the last few decades. Early methods measure the efficacy of antioxidants against the formation of particular species of oxidation products and are therefore based on lipid oxidation measurement. But today, a large group of assays are available for direct measurement of hydrogen atom or electron transfer from potential antioxidants to free radicals in simple lipid-free systems. Major antioxidant capacity methods can be classified into two. One, a hydrogen atom transfer method, which donates a hydrogen ion from a stable molecule, thus allowing the antioxidant to scavenge the ROS. And two, a single electron transfer, which depends on the potential of this antioxidant to reduce certain molecules and compounds by transferring an electron. This slide shows different types of methods which are categorized into one of these two methods, electron transfer or hydrogen atom transfer. Antioxidant activity can be monitored by a variety of assays that involve hydrogen atom transfer, single electron transfer, metal chelation, etc. Some of the common methods used are oxygen radical absorbance capacity or ORAC, 2,2-diphenyl 1-picaryl hydroxyl DPPH, ferric reducing antioxidant power FRAP, total reactive antioxidant potential TRAP, and cupric reducing antioxidant power CUPRAC. There is no simple universal method to assess antioxidant capacity accurately and quantitatively. Therefore, understanding the mechanisms and limitations of measurement assays is important for proper selection of method for valid evaluation of antioxidant potential. Epidemiological data suggest consuming diets rich in antioxidant nutrients and phytochemicals are beneficial to health. Dietary antioxidants have been isolated from fruits, vegetables, nuts, herbs, and spices. Dietary antioxidants have important roles in food or in human body. These compounds exert their activity by several mechanisms: free radical quencher, singlet oxygen scavenger, or chelators of metal ions. In the last 75 years, there have been several studies that explain the antioxidant profiles of different foods and their antioxidant potential. After discovery of antioxidants to prevent the food rancidity from lipid peroxidation, numerous antioxidant substances have been discovered and isolated from natural sources such as herbs, spices, vegetables, and so on. The slide here shows a timeline of major developments and milestones in food antioxidant research. As you can see, we have come a long way from food additive to design of functional foods. Next two slides show the antioxidant capacity of different types of foods reported as TEAC values. In the first group, there are different types of fruits. As you may know, berries such as blueberry and blackberry are considered superfruits due to the high content of antioxidants. The next group is vegetables, and they seem to contain relatively lesser amounts than the fruits. And the third group shows the much hyped beverages such as green tea and red wine. Our next group contains nuts such as almonds, hazelnuts, and walnuts. They are excellent source of antioxidants. Legumes and lentils are also rich in antioxidants. Pay attention to the high levels of antioxidants reported in rice bran. Now let us talk about spices, our topic of interest in today's lecture. Spices have received increased attention as sources of many effective antioxidants. Spices have many applications, namely as flavoring and coloring agents. They are used traditionally as preservatives. Spices are an integral part of traditional medicine, such as Indian Ayurveda and Chinese traditional medicine. The main components of all spices are mostly phenolic compounds such as flavonoids. For example, eugenol and cinnamaldehyde are present in clove and are related to antimicrobial and antibacterial activity. However, these compounds are not exclusive to clove. Cinnamon also contains cinnamaldehyde and possesses antimicrobial activity, but it also contains other chemicals such as spinnine, which confers antioxidant activity. The antioxidant compounds isolated from spices and their mode of action is summarized in this slide. Most of the antioxidants from spices act by reacting with free radicals created during the initiation stage of photooxidation. Others form complexes with metal ions. Now let us take a closer look at some of the common spices. As you know, a spice can be derived from different parts of the plant. For example, clove is the bud, cinnamon is the bark, ginger is the root, peppercorn is the fruit, cumin is the seed, and saffron is the flower. First spice in the list is rosemary. It is an aromatic evergreen shrub native to Mediterranean region. Rosemary and its extracts are used as natural food preservative. The principal antioxidant component of these extracts and the most widely studied of rosemary constituents are phenolic diterpenes, carnosic acid, its main breakdown product carnosol, and caffeoyl derivative rosmarinic acid. Further research on understanding this spice's potential to alleviate complications of obesity and diabetes, inflammation associated conditions, and neurological deficits are underway. Extracts from the leaves of rosemary are used as antioxidants in food, and a range of commercial products containing extracts of rosemary are available. These extracts are available in water dispersible or oil soluble forms. The second spice is ginger. It's a member of the Zingiberaceae family. Cell culture studies show that bioactive compounds in ginger, such as gingerol, showgol, and zingerone, have antioxidant properties. However, whether ginger antioxidant constituents are bioavailable in humans once ingested, and if they can affect markers of oxidative stress in human in vivo, are not yet known. Based on animal models and in vitro studies, ginger may have beneficial effects towards cardiovascular diseases through its multiple actions counteracting inflammation, hyperlipidemia, platelet aggregation, and hypertension. Moving on to cloves. Cloves are obtained from the dried, unopened flower buds of the tropical evergreen tree. Though bioactive compounds in clove have not been fully characterized, eugenol is believed to be the main contributor to its biological actions. Bioactive compounds in cloves have been reported to offer health benefits towards suppression of microbial growth, improvement of diabetes symptoms, and improvement of neurological problems. There is preliminary evidence that clove and its constituents can contribute to inhibiting oxidative stress, counteracting inflammation, and opposing processes associated with cardiovascular disease. But whether clove, when used as a spice in food, could have this health impact is yet to be determined. Turmeric is a spice that is derived from the rhizome of Curcuma longa, has been used in foods and in traditional medicine for centuries. Primary bioactive compound in turmeric is curcumin, which has potent antioxidant and anti-inflammatory properties and is thought to prevent cancer, Alzheimer's disease, heart disease, arthritis, etc. However, clinical confirmation of these benefits is limited. Another challenge is establishing the in vivo efficiency in humans due to poor bioavailability of this hydrophobic molecule. Improving the bioavailability of curcumin through development of new analogs and formulations or novel routes of administration would remove a major impediment to its widespread application and practical use. Moving on to cinnamon, it's derived from the bark of cinnamon tree and comes in two varieties, Ceylon and Chinese. It's one of the most commonly used spices worldwide next to black pepper. Cinnamaldehyde has been identified as the major constituent from the bark and eugenol as the primary component in extracts from the leaf. There is suggestive evidence that cinnamon can lower blood glucose and modulate insulin levels associated with type 2 diabetes. However, more research is needed to confirm these findings. Black pepper is the most widely used spice in the world. The chemical piperine is the major bioactive component present in both black and white pepper. It's been reported in animal models that black pepper and piperine can stimulate digestive enzymes, modify stomach secretions, alter gastrointestinal food transit time, and inhibit diarrhea. There are numerous cell culture studies demonstrating the antioxidant effects of black pepper and its extracts. However, its bioavailability once ingested by humans and the subsequent improvement in appropriate biomarkers in humans are not well characterized. Let's take a look at bioavailability. Not all of the micronutrients ingested gets absorbed into the body. Hence, to quantify the amount that is actually absorbed, distributed to the tissue, metabolized, and eventually excreted, the term bioavailability was introduced. Bioavailability describes the concentration of a given compound or its metabolite at the target organ. Food and Drug Administration defines bioavailability as the rate and extent to which the therapeutic moiety is absorbed and becomes available at the site of drug action. The pharmacological definition of bioavailability covers such linked and integrated processes such as liberation, absorption, distribution, metabolism, and excretion. Herbs and spices have been consumed for medicinal purposes to prevent or cure many diseases since the older civilizations. However, depending on the dose of exposure, the potential health benefits of phytochemicals in herbs and spices might be changed and reversed to toxic effects. These toxic effects include carcinogenic, neurotoxic, genotoxic, teratogenic, cytotoxic, nephrotoxic, and gastrointestinal toxic effects. For example, there are reports of adulteration of Chinese anise that was used to treat colic with Japanese anise, and that led to neurotoxicity. There are carcinogenic effects reported from different spices. Ginkgo biloba extract is a herbal medicine used for the development of memory and brain functions. According to a report published by National Toxicological Program, ginkgo biloba extract may cause cancers of both liver and thyroid gland in animal models. However, researchers are indecisive on this report due to the application of very high doses. Teratogens are compounds that may cause physical or functional defects in human embryo or fetus after the pregnant woman is exposed to substances such as alcohol or drugs. These are abortifacient herbs that may cause abortion when consumed in large amounts. So, what could have been the reason for such responses? Most effects of micronutrients in general, and dietary antioxidants in particular, follow a U-shaped curve with low or deficiency levels causing an increased disease risk. An optimal protective amount, which may be narrow or broad range, and an excessive level causing again an increased risk. So, in summary, free radicals and other reactive species are contributory factors in the development of chronic diseases. Herbs and spices are rich source of bioactive phytochemicals, and majority of these phytochemicals have powerful antioxidant activity. However, they do not contribute significantly to the nutrient makeup of our diet as they are used in low quantities. The functional properties are mainly attributed to polyphenolic compounds that are free radical scavengers, hydrogen donating compounds, singlet oxygen quenchers, and or metal chelators. Spices contain a cocktail of biologically active phytochemicals with diverse health benefits that have only begun to be elucidated. Animal and well-controlled human studies are needed to characterize dose-response relationships at culinary relevant levels. This concludes the lecture on spices and their antioxidant properties. Once again, it's been a pleasure sharing it with you. I would also like to extend my gratitude to the organizing committee for the opportunity. Thank you.